during and after docking. So if we talk about the pharmacophore modeling, could be classified into two different parts. It could be ligand-based or structure-based. So when we talk about ligand-based, in this category, we don't need any protein structure, but we need some active ligand structures that are already known. Similarly, in structure-based condition, we need some structures of target protein, and we can generate some models based on that structure, protein structure, and we can identify some drugs. So if we talk about ligand-based, for example, if we have some new emerging pathogens or diseases, for example, COVID-19, we are all familiar about. So in that case, we don't have any structure initially, but later we have. Similarly, if we have some novel proteins that we don't know what are the structures, there are no 3D structure or complex structure or co-crystal structures are reported on PDB, protein database. So what we can do in that case, we can go for ligand-based identification. But if we have some co-crystal structure of a protein in PDB database, then we can go for structure-based pharmacophore modeling. So in both cases, you can design some pharmacophore models, you can use AI or machine learning, and you can have some good molecules. So this is just the introduction part. We will discuss more. So if we talk about ligand, I hope you all are familiar with these databases. These are very common databases for chemical molecules where you can download the structure, you can study their smiley, their 2D structure, or their other property. Some of them are Zinc, Campbell, Drug Bank, RCSB PDB. So RCSB PDB is the data bank that I'm talking about. This can provide you the tertiary structure of protein, or you can say co-crystal structure of protein. Sometimes it also provides the ligand and protein complex that is very useful for pharmacophore-based model prediction or development. You can use UniProt, Malacard, and CVI. So these are some different databases that we are going to use for pharmacophore model development. For ligand, you can use Campbell or you can use Zinc. There are some natural database also, like you can download from Coconut or some other databases. If you talk about the target protein, you can download the structure from RCSB protein data bank. So using these two different approach, ligand-based and target, you can design your pharmacophore model. So for designing these models, you need different softwares, like you can use Schrodinger, HipHop, Hypogen, Disco, GSP, Phase, Discovery Studio, PharmaGist. So lots of online available tools are there, lots of download form or download version of software are there, and there are some paid or commercial software is there. For example, if you're going for Schrodinger, you have to pay some money, or you can request for 1 month free trial. If you mail the Schrodinger team, they will give you some 1 month or 1 week free trial. So in that phase, you can generate your own pharmacophore model. So after generating a pharmacophore model, you have to evaluate those models. So whatever your plan, either it is ligand or target based, you have to use your 80% data for model development and 20% data for training purpose. Once you design your model, you have to check it's like what is the sensitivity, what are their specificity, if it's working or not. So for that, you can use 20% of data to check if it's working or not. And based on that, you can use some other group of library and you can identify some good lead molecule. So these are some tools I'm showing here. You can use Ligand Scout 4.3. So you have to download this Ligand Scout, you have to install, and then you can perform your pharmacophore-based modeling on this platform. This is a very good platform. And besides this, you can also use Discovery Studio, MOE, and Pay. Here's the link of Ligand Scout download. You can download and install. It's very simple process. So once you open that Ligand Scout, you can easily upload your drug molecule and you will study what are their descriptors. Like you can also generate some own model based on these descriptors. So we will discuss more about this. In this class, I'm going to simplify these things in a protocol form so that you can perform your experiments and you can generate your own pharmacophore model and you can publish your own paper. So to simplify these things, I have written some steps here. You can perform step by step and you can generate your own pharmacophore model. So when we start, we need a specific disease so that we can select one of the receptors that we are going to target. For example, if we are using COVID-19 or SARS-CoV-2, SARS-CoV-2 is the pathogen, COVID-19 is the disease. And what we need a receptor. So we all know that there are lots of receptors that are involved in COVID-19 or you can say SARS-CoV-2 disease. If we talk about host, it could be ACE2, ACE2, or if we're talking about the pathogen, it could be spike protein. So whenever you are going to perform an experiment or you are going to design a project, you have to keep in mind if it's a pathogenic disease, you have to select either pathogen receptor or pathogen protein or host protein. So in both cases, you can select proteins. One of them could be receptor, one could be, or you can use as an inhibitor. Like, for example, if you're using for ACE2, you try to identify some drugs that can bind to ACE2 that cannot help to bind that spike protein to the ACE2 and it will inhibit that further infection of SARS-CoV-2. So this is just an example. But if you're working on a cancer, there could be different receptors. In that case, in case of cancer, we don't need any pathogen receptors. But there are some exceptional cases, like there are some viruses that are responsible for cancer. In that case, you can use the receptor of pathogen. But normally, in case of cancer, we use receptors from host cell. It could be CD36, it could be VGF. And there are lots of receptors out there. We can easily identify or we can easily find from literature. You can use database also. After that, we need to identify our inhibitors. So if you are working on structure based, you need to work on receptor. But if you are working on ligand based model, you need to identify some active inhibitors. So for those active inhibitors, you can go for literature search or you can go for some database. And once you have your known active inhibitors, you have to prepare your own library or you can use online libraries that are freely available to identify some unknown inhibitors, like they are not reported as inhibitors for that particular receptor. After that, we have to start making our list that could provide good features that can be further used for preparation of our model. For that, we use feature mapping identification. So these mapping identification will dictate or classify your drug model. It could be different type. You can generate 10 or sometimes 100, whatever you want. From a single molecule, from a single drug inhibitor, you can generate more than 10 models. And based on those 10 models, you will get different numbers of drug molecules. So if you're going to select some disease, you can use Malakar. If you're going to identify some receptors, you can use CADD pathway or drug bank. I will show you in the demonstration class how you can go to these databases and how you can select your receptor or disease for your project. So let's start with the feature mapping for pharmacophore model. Suppose you have identified your disease and you have your receptor or you have your a specific active inhibitor. Now what you have to do, you have to dictate or you have to write down some feature mappings, like what kind of features should be in that active compound. So for that, we have to generate or we have to identify some molecular pattern. Like we all know that any drug molecule is made up of carbon, hydrogen, oxygen, basically, normally. So in that case, there could be some hydrogen bond. So during hydrogen bond formation, there are some hydrogen bond donors and some hydrogen bond acceptors. There could be some cations or anions. There could be some aromatic compounds. There could be some hydrophobic features. And there are lots of combinations that you can create by using these molecular patterns. Once I'm going to show you the pictures, you will be very confident that, okay, you can create your own pharmacophore model. After that, we need to generate a 3D fingerprinting. The fingerprint can be used to analyze the similarity between molecules or among a library of molecules. Alternatively, a fingerprint model can be used to analyze the common elements of active compounds to identify the key contributing features to the biological compound. So every active molecules have a kind of fingerprints. And using those fingerprints, we can identify some similar compounds. The feature can be labeled as a single feature or any logical combination consisting of and, or, and not to combine different interactions patterns within one label. So just like computers binary language, there are some kind of, like whenever we give some kind of command to the computer, it store in binary language. Similarly, whenever we are giving some kind of features or molecular patterns, it's going to store like and, or, and not form. So this is the whole picture of pharmacophore fingerprinting. This will help you to understand how it works. So if you see figure A, in figure A, you can see these are the simple 2D structure of a molecule. Sorry, 3D molecule. And in B, you can see the 2D structure. So here in different colors, in figure B, if you see the figure B, the green color, small molecules, these are showing hydrophobic pattern. The orange one, these are showing aromatic. Like you can easily see that there are rings. So we all know that these rings represents aromatic. So orange means aromatic. If you see the cyan color, this cyan color means hydrogen bond acceptor. And the pink color of ball you can see here, this indicates these molecules are hydrogen bond donors. Now come to the figure C. In figure C, you can see easily that there are some distance between each molecules. The dotted line, you see the different dotted line between each molecules, like green balls, cyan balls. So there are some specific distance between these. If you come to the figure D, you can easily see there are some distance like D1, D2, D3. So these are all the features that we are going to generate in our pharmacophore model. And based on these distance, our computer generates some binary codes. For D1, D2, and D3, there could be different codes. And it's automatically generated by our system. So if you are interested to read more about this, you can go to Ching et al. Paper and you can read this. So here is example. You can see there is one drug molecule in 2D structure And when you design when you giving some features like acceptor, donor, or hydrophobic, and then in figure D, you can see there is different molecules with different distance. And once these features are designed, our computer is going to generate a model based on these parameters. Now, if we are going to generate our model, we have to generate some kind of queries. For example, we have a compound that has 6 hydrogen bond acceptor, 5 hydrogen bond donor, 4 anionic, 4 cationic, and 4 hydrophobic, and 5 aromatic properties. In that case, we can't go for all those parameters. We have to generate certain kind of queries, like which part of the molecule is interacting with the receptor. For example, if hydrogen bond acceptor is interacting with the receptor, this could be our main uh, feature. So based on that, we can reduce the number of features for generating our query. So here you can see that if a molecule can be fitted inside the sphere representing the query features, it is considered a hit molecule. Often, the pharmacophore query can be too complex to find hit molecules from a given library, and partial matching may be allowed. In such cases, only certain features considered essential for activity are matched. Additional uses of such model are to align molecules or facilitate molecular docking simulation. So this I will show you in my demo class, that how you can decrease the queries and you can get more number of hits. If you increase the number of queries, the number of hits going to reduce. So this is an example of pharmacophore model of BCL2 inhibitor, and this is generated by HipHop. It's software you can use to generate your own pharmacophore model. So here you can see there are different colors presenting different features, like hydrogen bond featured in green color and hydrogen bond donor in magenta color, ring aromatics featured in orange color, hydrophobic features in blue color, and others in black. So this is just a raw pharmacophore model. Nowadays, you can also use AI for pharmacophore modeling. This is a protocol, or you can see a diagrammatic view of pharmacophore modeling using artificial intelligence. Nowadays, there are lots of data generated by different research groups, and they are using database to store, and they're providing free to the users. So if you are interested to generate your own model, you can simply go to the database. You can download the huge data. There are huge data you can easily download and use those data to generate your pharmacophore models. So here it's a very latest paper. You can explore. It's a very good paper using artificial intelligence. They have generated pharmacophore model. What they have done, they have used some already known inhibitors. There are 46 known inhibitors for a certain disease, and they used to create 9 different clusters. Using different 9 clusters, they have generated different models. They perform some docking based, and they have some machine learning model. So using all these things together, they have generated a ligand based pharmacophore model. And using these ligand based pharmacophore model, they have identified lots of molecules, and then they use these molecules to perform molecular dynamic simulation for 500 nanoseconds. After that, they used these complex again as a filter, and they have generated a good complex pharmacophore model. Now they have ligand-based pharmacophore model, and they have also that complex-based pharmacophore model. So now they have generated a very good protocol that we can follow and learn more about how you can use not only ligand-based form for model, but you can also generate ligand and receptor complex form for models also. And whenever you are going to use your different library, you will get some good hit molecules. And that is not only for specific receptors. You can use different receptors or different group of libraries. So you can get good candidates. But again, there are some limitations of pharmacophore-based model. Like it is not fail-safe. Like there is possibility that you have found 20 molecules, and when you are going to perform wet lab experiments, there is possibility that 5 or 10 can be failed. So it's not fail-safe. And again, there is some absence of good scoring metric. It may be possible that the molecule cannot be identified if the confirmation is missing. And there is also possibility that we can get some good molecules, but there is also possibility that we can miss those active compounds. And the major limitation is that there is no one clear way to construct a pharmacophore query. You can just randomly generate your model. There are multiple combinations for your pharmacophore-based model you can generate. And we cannot fit all the properties of a drug molecule in a single parameter or a single model. Because there are lots of descriptors there are lots of biological features. There are lots of uh, complex. When you work on wet lab, in vivo or in vitro, and you work on computational work, both are very different. So this is not fail proof. There are lots of models that are working on some artificial cell based model, but it's still in early stage. Maybe in future we will have some uh, good model that can predict that, okay, if this drug is going to work inside human body or inside a cell, how it will work. It will be active or not. So here is an example. In many cases, kinase inhibitors are very similar to each other and yet have very different activity profiles for the kinome. And pharmacophore approaches aim at identifying kinase inhibitors would without any doubt identify kinase inhibitors like molecule. Nevertheless, there would not be a clear guarantee these molecules would be active for the targeted kinome. So these are the reference. If you are interested in pharmacophore modeling in virtual screen, you can read this paper. This is another platform you can use for molecular discovery. You can download, install, and you can design your own pharmacophore models using this platform. It's very simple. Once you download this platform, you just install it and upload your target protein or your target inhibitor that you are interested. So here you can see there is ligand-based option, and you can also go for structure-based for modeling. You have both the options here. So you can explore these things. And once you are done with this model generation, you can identify some novel drug molecules. And you can use also Schrodinger phase tool. This is very popular one. So this is a commercial software. So if you want to work on this, you just write a mail for 1 week free trial. They will provide you some password or some key for 1 week free trial. And if you have Discovery Studio, you can also use Discovery Studio for your pharmacophore model preparation. These are the working of all these platforms are similar. So if you have practiced with any one of these tools, you can work on any platform. FarmGist is a freely available platform. You can work on this also. Farm Mapper, you can work on this also. So this is the platform you can see. You just have to paste your, or you can also upload your file here. It could be MDL form or STF form or MOL form. You have to provide your email address so that once the job is done, you can get the details. And this is the Farmit server. You can use this also for ligand based or structure based model development. This is an example. In the left side, you can see the pink model. The pink structure is showing the receptor molecule, the 3D structure molecule, and the green represents the drug molecule. So you can easily see that drug is fit inside the pocket of that protein. So based on these structures, and in the right side, you can see different colors like green ball and yellow balls. So these are the pharmacophore features. It could be hydrogen bond acceptor, it could be hydrophobic. So these are different features. So once you decide your pharmacophore features, and you can find different hits. Less the number of features, higher the number of hits. If you increase the number of features, it's very obvious that you will get less number of hits because you are limiting the search criteria. This is another server, PGMG server. You can also use this server. So this is a deep learning based server. Right now, I think it's not working, but maybe in future, if they update, you can use this PGMG server, or you can simply mail them that you want to work on this server. So they will give you some link, and using those links, you can use this server. Once you use that server, you will get this kind of output. So you will get some output like you can see input pharmacophore. There is 5 aromatic rings, hydrogen bond 7, hydrogen bond acceptors, 2 hydrogen bond donors. You can also see different properties like quantitative estimator of drug likeness, what are the molecular weight, and accessibility. And you will have some good list of drug molecules. You can easily see their 2D structure here, their smiley form, and you can also see their 2D structure. If you click on the view button, you can easily see this. You can download in Excel form, and using these drug molecules, you can further go for like admit property prediction, docking, MD simulation, and some other computational approaches. So these are references that you can read to know more about pharmacophore models, how to generate deep learning based model, how to generate AI based models. These are very good papers. Thank you. Any query? Okay, if there is no query, I will move forward with practical session. So let's start with the protocol. I'm going to follow this protocol. So first step is selection of disease and specific receptor. So if you are not sure which disease you should select, you can explore Malakard. You can simply write Malakard or you can type malacards.org, or you can simply write Malakard on Google. You will see this page. This is the homepage of Malakard. It's a human disease database. If you are interested in pathogenic disease, you can write here. You have to write the name of pathogen. For example, if you're interested in Plasmodium, or you don't know the name of pathogen, you can simply write malaria. You see, it's a genetic disease, rare disease, and infectious disease. So, infectious disease means it's a pathogenic disease. You want to learn more about infectious disease, you can click here. You can read here. If you're interested in cancer, you can write cancer. There are multiple different type of cancer. You don't know which one you should take. You can simply come to this page. You can read here. Suppose you're interested in lung cancer, just click here. Now you can see the details of lung cancer here. And if you see on the left side, there are some quick search like summaries, trait, associated genes. Once you click here, you see there are 1400 genes that are associated with lung cancer. So there are different categories like RNA genes or protein coding genes. So whatever you are interested, you can just pick. And if you want to read more about drug and therapeutics, you can click here. You can see there are 7,674 drug and therapeutics records are in this here. See, there are 955 drugs for lung cancer are already reported. It could be in phase 1 or phase 2, 3 or different like phase 4 clinical trial. For example, if you see here amiodarone, you want to know more about this, like what kind of pathways or targets, you can go to drug bank.
Manisha, means is there a way out of these drugs which is, I mean, the ranking system which is the best one?
So we can use this phase 4. Like phase 4 is a very good molecule. This means that this drug is already past the 3 different clinical trials. So if it's phase 4, we can go for. Here in this table, phase
Madam,
4.
excuse me.
Yes, sir.
Phase 4 तो post market introduction है ना trial.
Yes, sir. It's a clinical trial.
After introduction to market, when authorities allow it for treatment. After that, phase 4 clinical
Yes.
trial.
So
Okay.
these are phase, like there are 3 different phases. First, second, and third. If a drug passed 3 different categories or 3 different phase clinical trial, then it goes for phase 4. So this means the phase 4 drugs we can use for our study to identify some new better drugs. So suppose there are 2 different drugs. One is in phase 1 clinical trial and another is in phase 4th clinical trial. So we only go for phase 4 trial because it's already passed 3 different clinical trials. If we compare, the phase 4 drug is better than phase 1 trial. Is it clear, sir?
Yes, ma'am.
Okay, so now we have one drug. If we go and search for drug bank, so here you can see in left side there are lots of quick search, and you can see targets. So before going to target, I want to introduce some other features also. Here what you can see the structure of that drug molecule, and you can see there are 3 different patents also, and you can also see their clinical trial. So you can see it already passed phase 1, 2, 3, and 4. There are different studies. The numbers are showing that these are different studies that cleared 16 different experiments cleared this phase 2 trial, and there are 43 different studies or experiments that cleared phase 4 clinical trial. So if you go for target study, you can see there are different protein groups. For example, here you can see HERG, human cardiac K plus channel. This is the one target. Another target you can see beta adrenergic receptor. A single drug have 8 different targets. So using these information, you can generate your own models. So in this case, if the structure is present, you can go for structure-based pharmacophore model development using single molecule with 8 different receptors. There is another protein group. Thyroid hormone receptor. It's a very common receptor. We all know about this receptor. And there are lots of drugs also out there. So if you are interested in identification of some drug molecules, you can search for other molecules also using these receptors. So we have lots of data. You can explore all these. Now back to this Mala card. So there are different pathways also. In this, you can see there are lots of genes here. EGFR, it's a very common receptor. We all are familiar with it. So you can explore these, or we have one more pathway database, that is CADD pathway. Here I'm taking example of malaria. So this is the pathway you can see, or you can also take other example. So here you can see different pathways. If we go with the first one, here you can see there is 2 different parts. The first part is pathogenic part, or you can say the SARS-CoV-2 virus part. And the second part representing the human cell, or you can say the host host part. So in first part, you can see the SARS-CoV-2 with S receptor S protein. So this S protein is going to interact with ACE2 protein of human host protein. So human is a host. So ACE2 is the host receptor that is going to interact with S protein, and it will help this virus to enter inside the cell through endosome, and further it will replicate inside the human cell. So here you can easily see that there are two different receptors or two different proteins, one from host, one from parasite. You can target these two receptors for your own pharmacophore model. So one thing is clear from this pathway, that you can easily find good receptors from CADD pathway or Malakard or from drug bank. If you are not sure what kind of receptor or what kind of target you want to work on. So these are the very basic and clear things that you can easily target. So if you're going to target S protein, you just use S protein structure. You can easily download S structure from PDB database, or you can use ACE2 protein that is also available on PDB database. And if you go for the PDB database or in literature, you can easily find there are lots of reported drugs, either experimental or computational studies, that these drugs are active drugs for S protein, and these drugs are active drugs for ACE2 human receptor protein. Now you have lots of data. You can generate your own pharmacophore model. This is another database, GeneCard, that you can use for identification of some receptors and what are their properties. You can explore and read about these things. So before starting your own project, you have to study very carefully about what are you going to target. Suppose you are targeting cancer. In case of cancer, you have to select host receptors. If you are going to target some other pathogenic disease or infectious disease like COVID-19 or SARS-CoV-2, in this case, you have two options. Either you can use pathogen receptor protein, or you can use host receptor protein. Now we have our receptors. We can also go for drug molecules. For that, you can go for PubChem. I hope you all are familiar with this PubChem database.
Yes, ma'am.
So here I am taking example of this molecule. So here you can see there are 69 compounds are there, but there is some modifications. Like this is amiodarone, but with hydrochloride. So someone has added this hydrochloride with this drug to increase its property or some bioavailability. Like if you add if you have an active compound and you add some other properties or other molecules, it will increase their bioavailability or their active compounds or some drug Like they can increase their drug availability, or it could be increase their inhibitory activities. So whenever there is some kind of modification, most of the time it increases its property. So you can explore these 69 compounds if you are working on this amidorem. And again, if you are familiar with this database, you know there is different proteins, different pathways. You can explore all these. You see, these are the human receptors that are interacting with this particular compound. Thyroid receptor, this same data we have on drug bank, but the drug bank is showing only 8 proteins, 8 target proteins. But here you can see 48 target proteins. You can explore these 48 proteins, learn more about their pockets, their binding pockets. So there is receptor. If there is receptor, there could be some binding pockets. So you can study more about the binding pockets of these 42 receptors, and you, based on those pockets, you can generate your own model. That model is going a part of structure-based pharmacophore model. So this is just an example for a single molecule. And if you want to explore about pathways, you can go for pathways. Action pathway, you can explore these things if you're interested. So now our first step is selection of disease and specific receptor is complete. Anyone has any query about this? I hope you all are able to identify a disease and receptor for specific pharmacophore model. Now go for second step, identification of already known active inhibitors with moderate activity using literature or database. So in literature, you can simply search on a particular active inhibitor. For example, as I showed here, amiodarone. You can use any other molecule also. Now you have your active inhibitor also. For your ligand-based pharmacophore-based model, you have your active ligand inhibitor. And for structure-based pharmacophore model, we have your structures. Now we have to retrieve the data. If you are familiar with PubChem, so this is the home page of PubChem for amiodarone. Here you can see the download. On the right side, you can see the download option. You can download 2D structure or 3D structure from here. And there are different forms like STF form, XML, JSON. So you can download from here. Or you can download from here also. You can also use this smiley form. So these smiley form you can further use for the prediction of admit property. So this is a soft tool to predict ADME for a drug. You can simply paste. So here you have two different options. Either you can paste your smiley form here on the right side, or you can draw your own molecule using Marvin software here, or you can also upload some new molecule. It could be 3D structure, or you can generate your own. So I'm just pasting one smiley formula here and you can run. So why I'm showing this ADME? Because if you are selecting your molecule and if it does not show any good ADME, then there is possible it's a waste of time. We should not take a molecule with bad ADME properties. So here you can see different physicochemical properties. It has 4 hydrogen bond acceptors, but no hydrogen bond donors. So you see it's a poor soluble compound. GI absorption is also low. No blood-brain barrier permeability, but it has good Lipinski. Only 2 are violated. There is 0 alert for pain. So these kind of properties you can predict using simple smiley formula that you can get from PubChem. Now, unknown inhibitor downloads. So if you want to generate your own library, you have to download some active compounds. For that, you can use zinc database or drug database. After that, we have to identify and list some features for your inhibitors. This is going to be your main part. For this, we can use different kind of platform. You can use Schrodinger, Discovery Studio. Here I'm going to use Farmit. So this is you can use PDB database to download the complex structure of receptor and drug molecule. So here you see 4UJ1. It's a PDB ID for an inhibitor for protein kinase A. This is a complex of protein kinase A protein receptor with inhibitor. So if you want to know about the inhibitor, you just scroll down. And here you can see chain A is showing protein kinase structure. And here you can see protein kinase inhibitor. And these are the drug molecules. So once you have option here to download this structure, go to download file and you can download either in FASTA sequence or PDF file. If you want PDF file, click on legacy PDF form. You will have your PDF form. And you can open this in FIMO. If you want to visualize, you can open this in FIMO. I hope you all are familiar with FIMO. So here you can see the green is the receptor part and the red one is the drug molecule. You can see this drug is found inside the binding pocket. So here the receptor is protein kinase A. I am just keeping the PDB ID here. You can simply type the PDB ID, and once you submit, you can see this structure. It's not clearly visible, so what we can do, we can hide the receptor part, and the background is white, so it's not very visible, so we can change the background. Now I think it's visible. So this is a simple example for a drug molecule. And if you see here, this is hydrogen bond. The hydrogen bond is presenting in white sphere. Hydrogen donor, sorry. The hydrogen donor is presenting here in white sphere. And the hydrogen acceptor is presenting in orange or yellow ball. And the hydrophobic feature is presenting in green sphere. If you want to add more properties like aromatic property, you can click here, and the aromatic property is showing here in purple color. And if you don't want to add any aromatic compound, you can simply switch off. You want to add 2 aromatic compound, you can add 2 more. Like it's all depends on your choice. It's free. Like it's on your free will. If you want to add more hydrogen acceptor, you can click here and you will get. Now we have these kind of properties. One thing I want to point out here, if you see this structure, these 2 aromatic rings, it's inside the pocket. These 2. So if we take 1 aromatic ring, it would be good, or we can skip this aromatic ring. For now, I'm not using this aromatic ring. Now we have our pharmacophore model with 3 different hydrogen bond acceptors. The 3 yellow spheres are showing 3 hydrogen acceptors. The 2 white spheres are showing 2 hydrogen donors, and 3 green spheres are showing hydrophobic property. Now we have 3 different features with different numbers. Now we have to select our library. If you go up to the search part, you can see there are different libraries. You can use Campbell, ChemDice, ChemSpace, Zinc. You can also use Zinc. But here I'm going to use PubChem for now. And once you click on search PubChem, this platform is going to search for all these features on PubChem.
Excuse me, ma'am.
Yes.
So these features are same for all the ligands, or are there any different types of features for different type of ligands?
Yes, it varies. It is
It,
different for different kind of ligands.
Depends on the binding affinity to the receptor.
No, that could be different. Suppose the same properties, suppose there are 5 molecules with same 5, whatever features you are giving here, those 5 molecules have same features, but the interacting or the binding site could be different. Those 5 molecules can share only these properties. For example, if you see these two yellow spheres, for example, these two spheres are interacting with the binding site. Now you have 4 different molecules that have these two spheres, but there is possibility that these parts are not going to interact with those receptors. Is it clear?
So suppose if this binding, this is not binding affinity, right, ma'am?
No, this is not a binding. This is just a structure-based screening, or you can say not a structure-based, pharmacophore feature-based screening. So here we are providing 3 hydrogen donors, 2 hydrogen acceptor, and 3 hydrophobic parts. So these are just features of pharmacophore.
So
You can add more features.
these are the essential features of a pharmacophore designing, ma'am?
Yes, these are essentials, but this can vary.
Okay, ma'am.
As I showed in that, if you see here, in this structure, you see there are 2 aromatic rings here inside the pocket, and these 2 rings are outside the pocket. So there is possibility that these 2 rings are involved in the interaction or binding with this receptor. So in that case, we can only focus on, okay. So here you can see there are only 6 seats if we are using that much features. So what we can do, this, and this. These 3 molecules are not the part of interaction with the receptor. So if we remove these part, let's see what happens. So just remember, we have 8 features here and we have only 6 hit. Now we are going to remove these 3 features. Okay, now you see there is only 2 rings here and we are focusing on this part of features only. And we click on search, let's see what happens. You see the number of hits are increasing. If you are planning a ligand-based pharmacophore model, use your molecule in PyMol and try to find which part of this molecule is interacting with the receptor. If you see here in the pocket, this is the outside and inside it's a pocket. So if you hide everything, you can see these two ring part, like this is the part that is interacting with the receptor. So if we are focusing on this part only, that we have provided the features. We have provided 2 hydrogen donor, 2 hydrogen acceptor, and 1 hydrophobic. No, 1 hydrogen donor, 2 hydrogen acceptor, and 2 hydrophobic. So in this case, you can see we have 73 lakh, 74, like more than 73 lakh hits we have. For more, like these are very lots of number of hits. So we want to minimize this number. What we can do, we can add aromatic ring here. So if we add here 1 aromatic property, now what are our query features? Our query feature is we want to identify some molecules that contains hydrogen, 1 hydrogen donor, 2 hydrogen acceptor, 2 hydrophobic feature, and at least 1 aromatic. And if we search, so number of hits will decrease. Once you increase the pharmacophore features, it will decrease the number of hits.
Ma'am, does it mean less the number of hits, it is more efficient?
Yes. Like more number of, like we have to balance both the things, like higher number of pharmacophore features, less number of hits. But we have to balance. Suppose you are increasing the number of features, in that case you will get less number of hits. So in that case, you have to decrease your number of features so that you can increase the number of hits. So we have to play with these 2 parameters.
Ma'am, one more question, like how can we validate in silico predicted structures to the real molecule that we synthesized? So how do we confirm it?
Can you repeat that question?
Suppose we are predicting structures through in silico model. So if we synthesize into a real molecule, so how can we validate the structure which is similar to the predicted one?
So you are designing new molecule?
Like this is just my hypothesis, like hypothetical.
Okay, in your hypothesis,
Mhm.
you want to generate new molecule, a novel molecule.
Yes.
So that novel molecule, you can use that novel molecule and you can upload those structures and you can easily see if it's superimposing or it's fitting inside your model or not. One thing. Second thing, you can use these models to check if your designed molecule has good admit properties or not. Because if your design mod, as I said, once you add something in your drug, it will change its physicochemical property and water solubility change. It could be in better way or it could decrease the value of your drug. For that, once you design your new molecule, check this admit property, and if it passes from here, you can use your model. You can use this model if the design drug molecule is the part of this, it's passing this features or not. For example, you generated a new molecule and it passed that admit test. Okay, if it's passed admit test, it's a very good chance that it can also pass in wet lab. But before that, you have to check if it's binding to your receptor or not. You can go for docking, MD simulation. So these are different steps you have to perform.
Ma'am, how do we identify off-target binding?
Okay. So you have new drug, you want to check if it's going to bind to your receptor or not, or any other hypothesis.
Yes, ma'am. No. Like suppose if there is a molecule and if I want to see whether it is going to bind to the target receptor only or is it going to affect some other receptors also?
Okay, it's a very good question. So in that case, you can go for docking. You can also go for some uh, grid-based docking. Like it will provide you some space that you can move your molecule and check if it's going to interact with that uh, binding pocket or not.
Ma'am, is there any tools that can automatically screen all the receptors available in a model species which we test?
Okay. For that, you can go for Schrodinger, or you can use yes, yes, you can use Schrodinger phase. This one you can use. If you don't have Schrodinger subscription, then I have to search for any freely available because I have never worked on that area. So I don't know if there is freely available structure based platform or not. But yes, you can use this platform for that kind of.
Okay, ma'am, because just because of different new topics are coming,
Yes,
so I am little confused. Like, are there any tools or softwares that can be used to screen any off-target binding of the ligands?
there could be, but I have never read about that. I have to look into in literature. But this is very good topic. I will definitely look into this.
Okay, ma'am.
Because most of the time we found one receptor structure and we use our drug molecules, like 100 or lakhs of drug molecules, to check if this binding inside the pocket or not. But
Mm,
it's a very difficult task to use different receptors, because receptors are very big molecules, proteins.
yes, ma'am.
So it's a very time consuming, and I don't think there is freely available platform available or not. But if you have Schrodinger subscription, you can work on that.
Okay, ma'am.
I will
Thank.
search if there is freely available, then I will let you know.
Okay, thank you so much, ma'am.
You're welcome. So now you can see, once you increase the feature, you will have 5 lab kits. Now we have one more aromatic ring here. So if you want to work on that kind of receptor based something, you can generate your own pharmacophore structure based model. I will show you here how you generate that in the same uh, platform. I will show you. For example, if we click on this one PubChem ID 1603152001, if you click here, you see this is the molecule. that is superimposing to our model. If you click on other molecule, you can see you can download all this information. You see, this is the long chain here. This molecule has this kind of property. So there is possibility that this molecule is going to act as inhibitor similar to your design molecule. So you have used one drug molecule to generate your Pharmaco model. And based on these features, you have generated one model. And in this model, you have more than 5 lakh hits. And this structure is showing that, okay, this molecule has this property, but it has lots of other molecules also. So what you can do here, you can also use hit reduction filter. Hit screening, you can also use hit screening. What you can do here, you can select the molecular weight. It could be whatever you want. So once you use these kind of hit screening filters, so it depends on your specific project. It varies project to project. If your binding pocket is big, you can increase the molecular weight. If your binding pocket is small, you have to balance the molecular weight. And again, you have to, like, you have given only few parameters, like only 2 hydrogen bonds, but it will also select all those compounds that has 4 hydrogen bond accepted. So in that case also you can minimize the number of fit by providing that, okay, if a molecule has less than 5 hydrogen bond acceptors and more than 2, and if hydrogen bond less than 5 and more than 2, this number of fit will decrease, because right now it's showing the molecule that is present on the screen, it's showing lots of hydrogen bond acceptor or hydrogen donor, lots of hydrogen bond. Like, you can decrease the number. It all depends on the project, what kind of project you are working. So it's all depends on your selection of parameters. If you want to decrease the hit screening number, you can use this hit screen filter. Here, another example, you can see this molecule is overlapping to your pharmacophore model, but if you look at the structure, it's very different. The features are inside your molecule. It's not at the C terminal or N terminal part. If you like, it's not in the starting or end part of the molecule. It's in the center of the molecule. So there is possibility it could be going to bind to your target, or it could not be going to bind to your target. For that, you have to perform docking, or you can use virtual screening. So it's a time-taking process. So in starting, I have removed the receptor part. So if I
ये फिल्टरिंग काम कर रही है क्या?
yes sir. सर अभी हमने हिट रिडक्शन स्टार्ट नहीं किया, हिट स्क्रीनिंग स्टार्ट नहीं किया.
वही देख रहा था, मॉलिक्यूल कम नहीं हुए.
So here are visualization options. You can visualize your receptor. I visualize my receptor now in cartoon form. And I have made it green in color, so it could be visible, clearly visible for all of you. So at the time of designing, you can hide your receptor. When you click on none, you can hide your receptor and you can easily work on your Pharmacophore model. Once you get your hits, you can select here, go to the cartoon, and you will have your receptor here. You can change the color also for better visualization. Here you can see these are the molecules. You see the number of hits, it's here. Similarly, if you change the number of molecular weight to 300, it will change. See the number of hits, it's different. So you can change the numbers by using hit screening purpose. And you can also see different molecules inside the receptor. You can visualize. It's not visible, you can change the background. Whatever makes you changes, like you can change whatever you want. After this, you have to download all the list and you have to perform admit test. You can use Swiss Admi or you can use admit 2. It's another tool for admit property. Sometimes a very successful drugs has bad admit properties. There are some very good drugs, but again, we have to balance our admit properties and selection of drugs. For example, there are lots of chemotherapy that are approved, but they have some side effects. But they're still working, but we don't have any options for chemotherapies. So we are using those drugs. They have some side effects. Still, we are using those chemotherapy drugs because we don't have any options, other options. But it's treating the patient well, so we are using those drugs. So we have to balance the admit property. So here you can use the admit tool. This is the home page. Here, you can perform admit screening from here. You click on get started. You can upload your file, the smiley list that you are getting from here. This list you can download, you can get your smileys and you can upload here and you will have your admit properties, all the drugs that you have identified from your pharmacophore-based model. So if you take one of the example from here, it will take time. So for now, I can just show you one example with one molecule. So here you just have to paste your molecule smileys form. Just copy. Submit. You see, the molecular weight is 645, and other properties you can see from here. So if you see here, the pain, it's showing 0 alert. It's a good signal. But if you look at the Lipinski, Pfizer and GSK, they are showing rejected. So this molecule is fail on the parameters of Lipinski rule, Pfizer rule, and GSK rule. And if you come to the toxicity, see it's a blocker of HERG, and it's a toxic. And the permeability, you can also see the distribution, but distribution is 100%. Blood-brain penetration is not here, it's not predicted. And the excretion part is here also. So you have to analyze all the parameters, and then you have to balance. So for now, we have our ligand, we have our receptor, we have our hits, like 360,000 hits are here. You can decrease these numbers by increasing the number of hydrogen acceptors. So this is just one example. You can use some other examples also. Six TUK. You can also use 6 TUK as an example. You can also use 41BD for your practical purpose. You can use these IDs for your practical, for your practice. If you have any problem with these things, you can contact me or you can ask me now. You see the number of hits decreased to 4,000 hits. So you can generate your model. You can play with the features and then you can get good number of hits. Now you can use these hits, go to the admit or Swiss model, Swiss admit tool, and you will have some good molecules. Then you can go for molecular docking, perform MD simulation. Once you're done with MD simulation, you can publish your first paper on pharmacophore model. It's very simple. It will take only 4 to 5 days to perform these experiments and 30 days to writing. And within 40 to 50 days, you are ready to publish your own pharmacophore model based paper. Any queries for now? Okay, so now let's talk about the receptor. Now I have another PDB ID for 4IBD. This is Ebola virus protein. You can download PDB file from here. So here in this structure, you can see there is water molecules you can remove. Molecules. can remove this small drug molecule also. We need only receptor, so we have to remove these molecules. So now we can export this structure. I hope you all are familiar with the tools that predicts the binding pockets. Can anyone suggest any binding pocket tool?
Binding pocket queen.
Binding pocket queen.
Sorry.
Any tool? A tool that can predict binding pocket inside that protein structure?
Autodoc.
Yeah, Autodoc is there, but any online tool?
Swiss doc.
It will take time. So Caspi is a tool that gives you instant pocket binders. I think it's not working now.
Hello, ma'am.
Yes.
Can I
Hello?
ask you a question?
Yes.
Yes, I want to. Yes, actually, I want to know that if it is the protein is PPAR gamma, can
Okay.
we generate a modeling with your that's whatever you are describing, na? From
So
pharmacophore
that's a
modeling
protein.
process,
Is
we
that a
can.
protein? Is that a protein?
Yes, PPAR gamma.
Okay.
Means it is actually the anti-diabetic. Actually, I am working on anti-diabetic. So
Okay.
PPR gamma, that is rosiglitazone and pioglitazone are two drugs available in the market right now.
Okay. You can work on
So
that
on
because you have PDB structure here. Because if you don't have PDB
okay.
structure, it's difficult to generate Funk4 model.
So-
But
yes,
once
yes,
you have,
ma'am. PDB drugs is there. Uh-huh.
then you can use.
Sir,
You can use.
okay, ma'am. Can you share? You are telling na the Actually, I am totally from medicinal chemistry background. So in your pharmacophore modeling, so the step you described, na ma'am, the step by step how we generate the model. So you please can give the step so we can generate our own with the desired proteins.
Yes, you
So
can
the
generate it.
steps actually I need to.
Okay.
Uh, Means a short
Like
flow
you
chart
have
type
your
if it is
receptor.
possible.
So you
Yes,
have
I
your
am having the
receptor.
receptor.
Okay.
Yes, ma'am.
So
The
you can
PPAR
just
gamma.
simply go to farm it. Simply go to farm it. You can see
Okay.
load receptor option here.
Okay. Load
You
receptor.
can load
Okay.
your receptor from here.
You can show the demo, no problem.
Yes, sir, I'm showing right now. Now you have loaded your receptor
Okay.
and you can go for search.
Okay.
Because this molecule already, like this receptor already has a molecule, you see.
Mm-hmm.
So based on this, first you search for some molecules and then you can generate your own features, like you can add aromatic rings or you can add hydrogen donors.
Mm-hmm.
So first you have
So,
to
ma'am,
start with
there
the
is a
lowest features.
there is a, you are describing, in this software, this hydrogen bond acceptor, donor, etc.
Yes.
etc. There are material.
Yes.
But I am asking, is there any substituted ring structure? Is any option in pharmacophore model generation? Means substitute groups, that is aromatic group is substituted, or
Yes, you
some
can add from here. There are lots of options. Once you add here, you can
Okay.
get to see here. You see? Here. For that, I have to hide
Remove the
this.
surface.
Sir, are you saying something? So now you can add any property that you want. Like if you want more aromatic ring, you just click here. You see there is one aromatic ring here. Now,
Okay.
whatever you are going to search, it will search
Mhm.
based on your given features. Whatever features you are giving to your pharmacophore model, this will going to search only those molecules that are going to fit on this model only.
Okay.
And we all know that there is a pocket inside a receptor. So there is possibilities that some of the molecule is going to bind, some are not.
But the substitution in a specific site, how we have to substitute in different group in a specific site of our that is means prospective drugs, means the ligand, means in your model the pharmacophore model is generated and the drug entity is attached with this, and that means I am asking that in that particular drugs, means in that particular ligand, not drug, in that particular ligand, if we change some, or if we alter some substitution, or if we addition some groups or some ring or something like that, so whether is in these parameters I am not that is it is not there, means in
I got
hydrogen
it. Okay. So you are planning to design some new molecules.
Yes,
and you want
yes,
to
yes.
check if it's in. Okay, for that you can load features from
The
here.
property of decreasing, that means I am developing some such other
Novel
model.
molecules. Okay.
Yes, new molecule. So that can fit in your model or not. That will be very helpful for us also, no? So that
Yes, yes.
the new entity, whether it will be more effective than the existing one or not. So something like that I want to know by your applying your pharmacophore modeling.
Yes, you can. So if I hope you are familiar with Marvin sketch. So
Yes.
for example, if you are drawing something here, just an example.
Or I can simply put the smiley also.
Yes, you can also put simply smileys here. No, but for pharmacophore model,
yes.
okay, let
So
me
new
explain
entity
again.
to add the new entity.
Suppose
Yes, yes.
you are using this as an inhibitor. Now you are designing some new. So for designing
Okay,
new,
okay.
suppose you have added some chain here
Yes, yes. Okay, I
and you
got it.
added some new benzene ring in this structure. Now you have
Yes.
making two changes
Yes.
and this is
And
new
accordingly,
smiley
I
feature.
get the smile also, na here. Accordingly,
Yes,
the
this is,
changes.
yes, it will change. The smiley will change according to this.
Yes,
Once
yes,
you
yes.
have your
That
smileys
that
here,
smile I can use for further
yes,
use.
now this is your smiley, this is your structure, and you can also
Okay.
save your structure from here. After that, you can run your admit properties if it's passing or not. If
Okay.
once you run, if the admit is passed, you can download this structure.
Okay.
After that, you can come back to this format. You see the feature, load feature option here.
Yes.
Once you click
Yes,
here,
yes.
upload that structure here. And
Okay.
after
Whatever the
that,
new
you
structure
can,
I can
yes, yes, you will get the pharmacophore feature of that newly discovered or newly designed drug or ligand molecule.
Very interesting. Okay, okay.
Yes,
So that can give the idea, na, to totally
yes,
whether
yes.
it is fit or not. Okay, okay. Thank you, ma'am.
You're welcome. It's still searching, maybe because of internet problem. Now here you can see this is the green is the receptor that you are using for now. And these are the different drugs that are showing hits for this specific receptor. You see all those receptors, sorry, the hits, ligand hits are going to bind in this pocket region. But again, this is just a possibility that it could bind. So for that, you have to perform docking and undisambulation to check if it's the binding is stable or not. Okay. For now, we have all the demonstrations that are needed to show from here. Okay, one more thing I want to show here. Before you are planning to generate your own pharmaco model, please go to that interaction part. Study the interaction part. Because if you know what are the binding pockets, you also have to know about the interacting residues. The interacting residues between receptor and the drug molecule. For that, you can visualize in PyMol. Just upload your PDB structure. And for better visualization, change the color of your ligand. Or you can use Discovery Studio if you have. It's
मनीषा
very easy to use Discovery Studio. Yes, sir?
मनीषा
Yes, sir.
ये एफ पॉकेट वेब भी यूज कर सकते हो, कास्ट भी काम नहीं कर रहा है तो
Yes, sir. Okay, sir.
एफ पॉकेट वेब. सेकंड नंबर वाला ऑफ पॉकेट एफ पॉकेट
Web.
वेब,
Okay.
हाँ ये वाला.
Yes, sir. So here you can simply add your PDB ID or you can upload your PDB structure also. Like here there is an option for load. I have PDB ID, so I will use PDB ID. So this is my protein structure. So this is a prediction web server for pocket identification. But right now I am just, it will take time. In meanwhile, we can see here that if you have Discovery Studio, use Discovery Studio. Right now in my laptop, Discovery Studio is not installed. So I'm just showing through PyMod. But if you have Discovery Studio, use Discovery Studio to find out the interaction between your ligand and your receptor. So if you select your receptor molecule, so steps, you can find fuller contacts or just intra-site chain or intra. It's difficult to see in this PyMod, but I'm just trying to show any one interaction if it's possible. It's difficult with PyMod, but if you have Discovery Studio, you can easily see what are the interacting residues of. Okay, it's difficult to find here, but if you have Discovery Studio, please go and find the interaction between these molecules. If you get to know that, okay, this region, this part of the ligand is interacting with the receptor, just focus on the pharmaco features of this part only. The first, like this first region. And always go for the head part that is inside the pocket. You see here the upper part of the ligand. The ligand that is upward, focus on the pharmacophore properties of that part, not the outer side ligand molecule. I hope it's clear for everyone. Most of the time, students make mistake that they are using the pharmacophore, they are focusing on the pharmacophore properties of the outer part, like downside of this ligand. No, we have to focus on the inside, like upper ligand part, this part, the upper part. Okay, it's still running. So you can explore this pocket web. It's very slow because of network problem. If you don't have that binding pocket, you can search for literature. And in literature, you will get some binding sites also. So whenever you are going for docking, always use binding sites and binding pockets. Most of the time, I have seen students are running their docking without using binding sites. They only prepare grid and they start their docking. Always use binding sites for better docking score.
ये चालू कास्ट भी सर्वर चालू हो गया.
Ma'am, how do we know the binding is temporary or permanent?
It's not temporary permanent. It's good or bad. Like you can use MD simulation and it's showing a proper stabilization at 50 or 100 nanosecond. It will be good. For now, there are commercialized binding affinity softwares are also there. So it's working now. Okay. So you can use this CASP to find the binding pocket. 4IBD. I'm using 4IBD. So here you can see these are the pockets. And these are the exact same pockets that we are visualizing here in the PyMod. So you will have different binding pockets. You see, there are different positions. So in this structure, there is only one pocket. So we can see only one pocket. But there are some receptors that has two different pockets. In that case, what you have to do, you have to check either in PDB that at which pocket your active drugs are going to interact to inhibit the property of that receptor. Because sometimes what happens, you see two different pockets and you are going to use the other pocket that is not associated with the inhibition of the activity of that specific receptor. So if you're using that pocket, it's useless. Before going to start with your study, you have to find out which pocket is active for that specific project. For example, that you have talking about that PIP receptor, there could be some different kind of receptors, 3MWH. There is possibility that there are two different pockets in single receptor. Here it's showing only one pocket. It's good that you have only one pocket. You can go with this one pocket only. I have worked on different projects, so I know sometimes there are three pockets. And those two pockets are useful for your activity and the third pocket is not. But when you are going for docking, there is possibilities that your drug is going to interact with the third pocket that is not useful. So you have to make sure that the binding pocket is active binding pocket that you are going to use for your docking. So here for your receptor, this is the binding pocket. This is presenting in red color. You can see. And this is the position like lysine 60, chain A lysine 60 is. And if you scroll down, you will have some other like leucine. So these are all the residues that are involved in the formation of that pocket. So whenever you are going for docking, make sure you select all these molecules on your receptor for docking. Okay. Any questions?